I'm studying leukemia, and the project that I proposed for this award was to identify molecular vulnerabilities in leukemia cells that have Ikaros dysfunction. The Ikaros gene is mutated in leukemia cells and seems to confer a poor prognosis for children with leukemia. In order to identify these molecular vulnerabilities, we have used a genome-wide shRNA library. But first, to do our big experiment, we really had to develop the model system that was adequate to look for these molecular vulnerabilities. So we have created a pro-B cell line in which we can induce the dysfunction of Ikaros. And now we have transduced that cell line with our library and have performed the experiment. And now we are working to validate the results of our large-scale experiment. So when we do these large-scale experiments, we generate a lot of data. And so in the coming months and year, we will wade through those data. And in our lab, we take a very translational perspective on the results. And ideally, what we will do is identify targets that we can inhibit, ideally therapeutically, for those patients whose leukemia has Ikaros dysfunction. Being a CureSearch young investigator has really helped guide the focus of my research. I did a lot of research in acute myeloid leukemia before, but this award was for acute lymphoblastic leukemia. And so it really allowed me to change the direction of my research.